Hello, biology students, and we are going to do chapter 10 today, which is on meiosis and sexual reproduction. Now, this is important because this is the way that our bodies are able to make a diversified organisms, making them so they're not genetically the same as the parent. And so this is going to happen in the gametes of the body and create unique individuals. So this will cover overview of meiosis, genetic variation, the phases of meiosis compared to mitosis, cell cycle, and changes in chromosome number and structure. So one of the reasons that this is important is it introduces diversification. So as we've mentioned before, the amount of diversity is extremely important. You don't want everything to be the same. You want them to all be different. The more different kinds of things available, the better, because that means that it's a better chance of an organism being able to survive. So there are more than 70 trillion different genetic combinations possible from the mating of two individuals. So that is a lot of different combinations, and we see these combinations become possible during the meiosis process. Males and females differ in the way they form gametes. Gametes are the reproductive, basically the egg and the sperm, if we're talking about animals. Now in plants, that's the pollen and the ovules. In males, the sperm production begins at puberty, but in the female, the process of producing eggs starts before birth and ends at menopause. So this is only used for sexual reproduction. So if an organism is not reproducing sexually, then they have no reason to go through meiosis. The parents are diploid. We've already mentioned that. And in humans' case, that's 46. So you have 23 from one parent and 23 from another. What happens with meiosis is you get something that's haploid, or 23. So an egg has 23 chromosomes. A sperm has 23 chromosomes. When you combine the two together, you get the total of 46. So it's a combination of the two parents' genetic information that's going to make one full set of information for that offspring. The gametes are the egg and the sperm fuse during fertilization, and they form a diploid zygote. That's the original egg that is fertilized and will eventually develop into embryo. If events in meiosis go wrong, gametes contain the wrong number of chromosomes. So we see this most commonly in Down syndrome. Down syndrome is called trisomy 21 because they have an extra chromosome at the 21 chromosomes. So they actually have 47 chromosomes instead of 46. There are cases where individuals can have less chromosomes than 46. A lot of times these mutations will result in termination of the pregnancy, just spontaneous termination. But in some cases, like with Down syndrome, the offspring does live longer. So in diploid body cells, chromosomes occur in pairs, and a human, of course, has 23. So you have one chromosome that codes for eye color from your mom and a chromosome for eye color from your dad. One for hair color from your mom, one for hair color from your dad. And the way these two combine and work together is what determines what you actually look like. Homologous chromosomes means that they are similar in appearance and size and structure. They code for the same things. So you have a homologous chromosome, one from mom, one from dad, and those two homologs are going to match up during the meiosis process. Same length, the centromeres are in the same position, and they show similar banding patterns. So homo means the same. Homologous chromosomes means we're looking at the same chromosome pairs. So here's just an example. 23 pairs, 22 pairs plus the chromosomes here for the X and the Y chromosome. In this case, because there are two X chromosomes, that means that this individual is female. X and Y would mean that it was a male. So you can see that there are 23 pairs, including the XY chromosomes, or in this case the XX chromosome, and they have centromeres that are in similar positions, and they have banding that is similar too. So this is called a homologous pair of chromosomes, one from mom, one from dad. Now oftentimes we can't tell which is which unless we look at the parent genetics too, and then sometimes we can match up and figure out, okay, this one's from mom and this one's from dad. But a lot of times it's very difficult to tell, especially if you don't have the parent chromosomes to compare it to. But each one of these you can see have a similar appearance, and these, one from mom, one from dad in each case. So here we have a chromosome. Here we have a sister chromatid, keeping in mind that as we learned in mitosis, this side is genetically the same as this side. So this is two exact copies of our original one over here. But we also see that happen with the male or the female. So let's say, for instance, this is the father, the paternal, and this is the mother, maternal. So the mother has one chromosome that has duplicated into sister chromatids, and the father has one chromosome that has duplicated into sister chromatids. Genetically the same, genetically the same. However, these two, even though they're homologous, they're genetically different, but code for the same information. So homologous chromosomes have kind of the same codes, and we will call these alleles. Alleles are alternative forms of a gene. So you can have an allele for blue eyes versus an allele for brown eyes. And the way these alleles work together will help determine your overall eye color. Now, these alleles are found in similar locations on the chromosome, and we'll get more into that when we talk about genetics and get more into genetics in more detail. Identical alleles for a specific gene on both homologous are called homozygous. So let's say you have blue eyes from mom and blue eyes for dad. That's a homozygous trait. If you have blue eyes from one parent and brown eyes from the other, because the two alleles are different, we call that heterozygous. So homozygous means the same, so like blue eyes and blue eyes. Heterozygous means you've got a blue eye allele and you've got a brown eye allele, so two different kinds. So a gene coding for short fingers on one homologous and a gene coding for long fingers is the same location on the other. So basically it's saying that it's on the same chromosome, it's kind of in the same place, but one says short fingers and one says long fingers. In that case, because they're two different traits, it would be heterozygous, hetero for different. Now, there's two parts of meiosis. There's meiosis 1 and meiosis 2. The whole point of dividing twice is that we get half the information that we need. So in mitosis, we divide once and we get a full set that is identical to the parents. So in mitosis, we start off with a somatic means body, a somatic cell, and that somatic cell is going to divide and make an exact copy of itself with the exact same information. What we see in meiosis is in meiosis 1, the cells are going to divide one time, but we still have too much information, so the cells are going to divide a second time, which is meiosis 2. And by the time meiosis 2 is completed, we will have 4 cells that are a result from the original 1 cell. And each of those 4 cells will have half of the information necessary to produce an offspring. So in meiosis 1, the chromosomes are replicated because remember, replication takes place in the S phase of interphase. Each chromosome consists of 2 identical sister chromatids because they have replicated and they've joined together and they've got those 2, that X kind of appearance. The homologous pair up in synapses. Synapses are kind of the joining area where they join together. Homologous pairs align themselves each side by side. So this is what's important in the meiosis 1. It's different than what we saw in mitosis. In meiosis 1, we have the homologous pairs match up. So eye color from mom matches up with eye color from dad. That's what we see happening in meiosis 1. In meiosis 2, we see the sister chromatids that are going to separate. So in this case, we don't have homologous pairs anymore. They were separated out in the first meiosis 1. In meiosis 2, it's much more similar to mitosis in that the sister chromatids are going to get separated into different cells. So the best way to really look at that is to take it step by step, because I know it can sound confusing, although this slide is a really good summary of the 2 processes. We can see here that we've got our basic animal cell and we've got our homologous pairs. In this case, we're going to keep that the red is from the mother and the blue is from the father. In a typical cell, we've simplified it here to just show 4 chromosomes, just to keep the simplicity of it. But remember, this is happening for 46 chromosomes within a human. So we have the male and the female, and they have, here's a homologous, these 2 long ones, and these 2 short ones are also homologous. But we'll see the nuclear membrane start to break away during meiosis 1, prophase 1. So the DNA has replicated itself here. Remember, this happens in interphase, S phase of interphase, and the DNA has replicated. So we have duplicate of the same chromosome, another duplicate, and another duplicate, because these are sister chromatids. So with the DNA replication, we see these spindle fibers start to form with the centrioles. Remember, centrioles only appear in animal cells, with the spindle fibers between them. Throughout the first process of meiosis, we will see that the sister chromatids get separated out by their homologous pairs. Basically what this means is we want one big and one little in each of the resulting two cells. We don't want too little over here and too big over here. We don't want 3 over here and only 1 over here. That's what causes genetic mutations. We want one big and one little over here and one big and one little over here. Now there are different ways this can be combined. Maybe both blues end up on that side, and that's okay. That's part of the random process. Maybe both reds, one big red and one little red, wind up over here, and that's also okay. Another part of the random process. You can see how this would be very random if we had 46 chromosomes that we had to work with. So the first step, meiosis 1, is where we take the homologous chromosomes and we separate out the homologous ones. So one big and one little on each side. The problem that we see here, though, is that it's still replicated. They're still in their sister chromatid forms. So we have exact copies of this little red one, and we have exact copies of this bigger blue one. That's still too much information. So then meiosis 2 happens. And during meiosis 2, we're separating out these sister chromatids, just like we saw happen in mitosis. So this process is much more similar to mitosis. And it's going to result in four cells that are not identical. 4 cells that are different from the original. The original was a diploid, and these resulting ones are actually going to be haploid cells. So they each have half of the information. So this one had 4 chromosomes, 2 big, 2 small. These resulting cells are going to have 2 chromosomes, 1 big, 1 small. All right, now there's other ways that genetic variation can take place. One of them is crossing over. So with crossing over, basically what happens is those homologous pairs get together and then they actually switch some of their genetic information. So they cut off a little piece of themselves and they switch it with their homologous pair. This is great because it means that you can get something completely different that you haven't observed before. It can also cause problems because if you're actually cutting off a piece of that chromosome and switching it, there's a lot of room for mistakes that can be made. But that is one of the ways that genetic variation can happen. So it is a very important process, this crossing over. Another way that genetic information can be shuffled around is by independent assortment. That means that when the homologous chromosomes line up along the metaphase plate, or they're in the center, that they can get separated and pulled out randomly. And we kind of talked about it when we looked at this, that we could end up with 2 blues on this side, or we could end up with a big blue and a little red. As long as there's a big and a small on each side, it could get separated out randomly. So the way that they line up right here determines how they get pulled apart, and that's another way that the chromosomes can get shuffled randomly. So this is called independent assortment. So with crossing over, it's involving actually exchanging genetic material, and a little chunk gets broken off of one homologous chromosome and it gets switched with another one also broken off. The homologous chromosomes will be separated later on, and that is where another assortment can take place. So here we can see that crossing over that takes place, keeping in mind that this process occurs during meiosis 1. We've got 2 chromosomes that are homologous. In this case, we've got a mom one and a dad one, and they are similar in size and structure and appearance, just like we talked about before. But here you can see that they are crossing over with each other, and this genetic information is getting flip-flopped around. So now we've got a chunk of blue here and a chunk of red here. And that means that these are completely different chromosomes than what we had before. Again, a very, very important process because this allows for brand new genetic information that we didn't see before. Most of the time this is good, sometimes it's bad, sometimes it's neutral. We don't even notice a change. But this crossing over is an important step to mixing up that genetic variation. All right, so when homologous chromosome pairs align at the metaphase plate, we can have what's called independent assortment. Independent assortment means that it goes to different places randomly and they separate in a random manner. So that means the maternal can go to one side or the paternal could go to the other side. This causes a mixing up of the genetic information. So again, we have very different results. So here we can see some different combinations, and this is just using 3 different chromosomes as examples. Again, thinking of the red as the mom and the blue as the dad, and that these are homologous that have already replicated. So they could line up with all the blue on one side and all the red on one side, or we could get this combination or this combination, and you see that there is a possibility of 8 different combinations just from looking at 3 different chromosomes. Now you can imagine with 46 chromosomes, the amount of possibility during independent assortment is amazing. It's very random how they line up on this metaphase plate and which side they fall on. So when meiosis happens, meiosis 1 happens, all of these are going to get pulled to one side. The sister chromatids stay together, and instead the entire sister chromatid gets pulled to the side. And that's the results that we're seeing. All of this getting pulled to one side, all of these getting pulled to one side. And then during meiosis 2, that's when we'll see the sister chromatids separate out. So fertilization is the union of the male and the female gametes, and chromosomes donated by the parents are combined. In humans, there are this many chromosomally different zygotes are possible. So what is that? Million, billion, trillion? That's a lot. That's a lot of different combinations that are possible for genetic variation to take place. If crossing over occurs only once, then we can see there are this many genetically different possibilities. I'm not even going to try and figure out how many numbers that is. But that's if crossing over only takes place once. And crossing over can happen amongst any of the 46 chromosomes, although typically it's going to happen on the chromosomes that are a little bit larger or longer because it's easier for crossing over to break off a piece and switch it. Crossing over may occur several times in each chromosome. If the chromosome is long enough, maybe there are several places that it can cross over. So genetic variation is very, very random and very important, as we've mentioned. And this allows for better survival of a species as a whole. Now, that is the advantage of sexual reproduction, is looking at the survival of the species on a grander scale. It's not so much the individual survival, it's the species as a whole survival that sexual reproduction allows for them to experience. Asexual reproduction produces genetically identical clones. So if a single-celled creature just makes a clone of itself, well, that's great. It doesn't take as much energy. The organism doesn't have to find another organism. It doesn't need another individual to mate with. It can just reproduce itself. But if there's something genetically wrong with the individual, then it just made a copy of those same genetic mistakes. So that's, again, a reason why sexual reproduction is important. In humans with 23 pairs of chromosomes, the possible chromosomal combinations are 2 to the 33rd, assuming no crossing over has occurred. Asexual reproduction is advantageous when the environment is stable and when it might be difficult for them to be able to find another organism similar to them. However, if the environment changes, genetic variability is more of an advantage because some individuals will be better at surviving those adaptations than others, and the ones that do survive can pass on those good genetics to the next generation. So the phases of meiosis, we have meiosis 1, and it goes through prophase, metaphase, anaphase, and telophase, but we call it prophase 1, metaphase 1, anaphase 1, telophase 1 because it's all in meiosis 1. The steps are very similar. So prophase 1, the spindle fibers form, the nuclear envelope breaks apart, and the nucleolus disappears. Then we have the chromosomes are duplicated, which we saw in the interphase portion. The homologous chromosomes are going to start to pair up, and this is also when crossing over is going to take place. The homologous are referred to as bivalent, or as a tetrad, because it's forming 4 chromatids, whereas in mitosis they just form sister chromatids. In metaphase, the homologous chromosomes are arranged randomly at the metaphase plate, and they are attached independently to the different spindle fibers because the homologous chromosome as a whole gets separated out during anaphase. So each homologous chromosome that has 2 sister chromatids attached to get pulled entirely to one of the poles. The sister chromatids do not separate, and this is why we need to go through another division process because we still have too much information. Yes, we separated out the homologous chromosomes, hopefully, if everything went the way it was supposed to, but we do have too much information and we need to separate out those sister chromatids. We do see telophase 1 happening, where the daughter cells have 1 unduplicated chromosome from each homologous pair. Then we have interkinesis, which is similar to mitotic interphase, except it is usually shorter and the DNA replication does not occur here. This is where we now have 2 cells that are a result of 1 cell, but again, too much information, so we have to start the process again with meiosis 2. With meiosis 2, this process is similar to what we saw in mitosis, where we have prophase 2, metaphase 2, anaphase 2, and telophase and cytokinesis. So in prophase 2, the chromosomes are going to continue to condense, and they align at the metaphase plate. They're no longer in homologous pairs. They just align regularly like we saw in mitosis. The chromosomes get pulled apart and in anaphase 2 and separated out to the 2 poles. Then we have telophase and cytokinesis take place, and from 1 original cell, we now have 4 genetically different daughter cells that are formed. These are genetically different from the adult. They're haploid because they have half of the information that the original parent cell had. So here we see an original comparison of meiosis 1 and then meiosis 2. So meiosis 1 here is our gamete. It goes through meiosis 1 process where we've had crossing over take place. You can see a little bit of color variation there representing crossing over. The homologous chromosomes get separated out because we have the big one and the little one lining up randomly on each side. So we have a big and little on this side and a big and little on this side. They get pulled apart and the cell divides. Then we go into meiosis 2, which again is more similar to the mitosis process. And we have those get separated out until we eventually end in 4 genetically different daughter cells. So you can see here that's different than the original cell we started out with. And we've had 2 division processes to get to this point. This is another one of those good slides that you may want to pause and take a screenshot of because it spells everything out step by step, and it would be really helpful for you to study a slide like this. So here we have our meiosis 1 process, and here we have our meiosis 2 process, the step by step that's going on in each. Alright, so here's a side-by-side comparison of meiosis and mitosis. And meiosis requires 2 nuclear divisions, and mitosis only requires 1. Meiosis, the chromosome synapses and crossing over, there is no crossing over in mitosis. Centromeres survive in anaphase 1 because the sister chromatids are still joined together. Halves the chromosome number, produces 4 daughter nuclei, and they're genetically different from the parent and only used in sexual reproduction. With mitosis, 1 division, no synapse, chromosomes do not synapse or cross over, centromeres dissolve, and the chromosomes, sister chromatids, do separate out. 2 daughter cells are produced, and they are genetically identical to the parent and genetically identical to each other, and used for asexual reproduction, but also for growth of an individual because you're making exact copies of the same cell. Meiosis 1 and mitosis similarities. An orderly series of stages is involved. The stages include prophase, prometaphase, metaphase, and telophase. The spindle fibers play a role in sorting the chromosomes, and cytokinesis follow the end of process to divide the cytoplasm between the daughter cells. Again, side-by-side comparison of meiosis 1 with meiosis 1 with mitosis, including prophase, and we're seeing a difference of crossing over taking place in meiosis 1. And also, again, another main difference is the homologous chromosomes are what are being separated out, not the sister chromatids. But if we compare mitosis with meiosis 2, we see a lot more similarities here. Even though we're making haploid cells in meiosis, we do have the sister chromatids being separated out, 4 daughter cells being produced in meiosis, whereas mitosis, we're still getting those 2 daughter cells that are the same. Side-by-side comparison of the two processes, making sure that we understand that here's our main difference, crossing over has happened, and the homologous chromosomes are getting separated out, whereas here, the sister chromatids are separating out. A second division takes place in meiosis, where we end up with 4 daughter cells, and only 2 daughter cells are produced in mitosis. So the life cycle of a cell is all the reproductive events that occur from one generation to the next similar generation. In plants, they can sometimes have what's called haploid multicellular individuals that alternate with diploid multicellular individuals, and we call this alternation of generations. So basically it means that at some stages they may have haploid cells, and at other stages they may have diploid cells. The haploid cells are called gametophyte, and then the diploid cells are called sporophyte. And they actually kind of sometimes, depending on the plant, go back and forth between these two. And this allows for this alternation of generations. The diploid individual may be larger or smaller than the haploid individual. And mosses are haploid for most of their life cycle. In fungi and most algae, only the zygote is diploid. Ferns and higher plants are diploid for most of their life cycle. Again, keeping in mind that diploid means a full set of genetic information, so in humans, 46, and haploid means half the set of information. In plants, algae, and fungi, gametes are produced by haploid individuals. So it's a little bit confusing, but what it's saying is that it's a little different in plants, and some stages of a plant's life may only have the haploid phase where only half the genetic information is present. Now in animal cells, we're going to see meiosis occur in gametogenesis, which is the formation of the gametes. So that is the egg and the sperm process. In sperm, it's called spermatogenesis, and all 4 of the resulting cells, remember meiosis produces 4 cells, all 4 of those are going to become sperm, which makes sense when you think about it, because when there are sperm present, there are usually millions of sperm that are released in one ejaculation, and so those sperm are a result of the meiosis process when all 4 of those haploid cells become a sperm. Now, for the production of an egg, we call this oogenesis, and 1 egg is formed, and the other 3 are called polar bodies. And the polar bodies have different functions that kind of assist the egg through the process. But 4 cells are still formed, but only 1 of those is going to go on to become the egg. The other 3 will become polar bodies. So the sperm and the egg are produced by meiosis, spermatogenesis, and oogenesis. Sperm and an egg fuse at fertilization. This results in the zygote, the one-celled stage of an individual of the next generation, which undergoes mitosis. So when that egg and that sperm first fuse, and it is completely fertilized with a full set of information, and it's still one single cell, it's called a zygote. As it continues to divide, that 1 cell becomes 2, those 2 cells become 4, those 4 cells become 8, and so on. Eventually, we're going to see that number continue to double and double and double in order to make an embryo. It takes a little while to get to that point, obviously. And the results of this is because of mitosis, because the original cell is going to go through the mitosis process to grow. So again, in spermatogenesis, the testes contain stem cells called the spermatogonia. Spermatogonia make primary spermatocytes that undergo spermatogenesis. All of that to say, you get sperm this way by going through the meiosis process. Primary spermatocytes undergo meiosis 1, form secondary spermatocytes. Secondary undergo meiosis 2 to form the spermatids. Spermatids will go through a little bit of a changing process that's going to form the sperm. Now in oogenesis, again, this is different. It's going to happen in the ovaries, which also contain stem cells that called oogonia, and they produce primary oocytes. Primary oocytes begin oogenesis, but only a few continue at sexual maturity of the female. So we have the primary oocyte forms a secondary, and then the polar bodies. The secondary begins meiosis 2 but stops at metaphase and leaves the ovary and enters the uterine tube. If there are no sperm in the tube, the secondary oocyte degenerates. And if there are sperm in the tube, it triggers a secondary oocyte to complete meiosis and another polar body forms. So fertilization actually takes place within the uterine tubes. It does not take place within the uterus. By the time the fertilized egg implants into the uterus, it has already been fertilized for a while. It takes a while for it to actually travel down the tube, and that means the sperm actually has to travel through the uterus into the uterine tubes in order to fertilize the egg. All right, so here we can see how the process works. The male produces the sperm through spermatogenesis. The female produces the egg through oogenesis. The two are fertilized, making a diploid. So the egg is haploid, the sperm is haploid. The two are fertilized to make a diploid. And then that is going to produce a diploid organism. And then, of course, the cycle is going to repeat once the organism develops and grows. This is just showing kind of how the process works. So we've got our original primary spermatocyte, and it's going through mitosis process. When we get our spermatids, which have half of the, here we can see the N representing our haploid. Remember, N is haploid, 2N is diploid, resulting in 4 mature sperms. Then we have oogenesis, and with oogenesis, we can see here is our first polar body and our second polar body, and eventually, if the sperm is going to form, or sperm is going to fertilize it, we get our full set of genetic information. Now, sometimes this can change chromosome structure if the chromosomes don't separate out the way that they're supposed to. Euploidy is the correct number of chromosomes in a species. So this is what we're aiming for, and this is what, when everything goes right, we experience euploidy. In science, word U means true, so U is usually a good sign. Aneuploidy is a change in the chromosome number, and sometimes we can have too many, and sometimes we can have too few. If a karyotype is a karyotype is a display of the chromosomes arranged by size and shape. We saw that kind of before. I was seeing if there's another picture of a karyotype. But a karyotype is where you have all the X's, the chromosomes laid out nice and neat beside each other, so you can compare which came from which parent. Sometimes a karyotype will show that there are too many or too few chromosomes present, and a lot of times this results from non-disjunction or the failure of the chromosomes to separate. So if you think back to when we have independent assortment, where we have the same number of chromosomes on each side in metaphase 1 of meiosis 1, we have the homologous chromosomes paired up. Well, what happens sometimes is that the homologous chromosomes don't go to their separate cells like they're supposed to. One of them may actually pull an extra chromosome into its cell, and that's going to result in too many chromosomes in a couple cells and too few in another. So monosomy is only one of a particular type of chromosome is present, so that you're missing one. Trisomy is that there is an extra, and as I already mentioned, one of the most common ones is Down syndrome, which is trisomy 21. So that's an extra chromosome, tri meaning 3, so they have 3 chromosomes instead of 2 at the 21 juncture. So here we can see an original one, this is what it's supposed to look like, but instead of these getting pulled apart like they were supposed to, like we see here, they got stuck together and pulled into one cell. So now this one has way too much information, and this one over here has barely any. So this one is going to be monomy, and this one is trisomy because it's going to end up with extra information. So again, these two are going to end up with not enough. These two are going to end up with too much. And we can see this happen in a normal cell that goes to a normal cell, and then disjunction, non-disjunction happens in one of, or it could happen during meiosis 2 as well, and so meiosis 2 can also result in that, in the wrong number of chromosomes. But basically it happens when they don't get pulled correctly to the correct cells. So in this case, we end up with some sperm that don't have everything they need, and in this case, we end up with some eggs. This one's got too much, this one's got too little, these two have too much, and these two have too little. So we're missing information here, and we have too much information here, and these are the only ones that had the correct amount. So again, trisomy 21, I know I've already mentioned that, is Down syndrome. It is where there are three chromosomes at the 21 chromosome, because remember at the very beginning we looked at pairing up those chromosomes in a karyotype. And there were 22 different chromosomes, with the 23 pair being the X and the Y chromosome. The chance of a woman having a child with Down syndrome increases with age, so usually 35 or above if a woman is 35 or older and has gets pregnant after 35, then there's an increased risk of Down syndrome occurring, which means that the eggs did not separate the chromosomes properly. This is characterized by kind of a shorter stature, eyelid folds, a flatter face, stubby fingers, and wide gaps between the first and the second toes. Here's a karyotype. I know I've been referring to those. This is what a karyotype is. We have the chromosomes that are matched together, one from one parent and one from another, and we looked at this at the beginning of this outline. The 23 chromosome is right here, so this is the X and the Y, so this tells us that this was an individual. Also, for future reference, you might notice that the Y chromosome is much smaller than the X chromosome. That's because the Y chromosome doesn't carry a lot of genetic information, usually just enough to say that it's a boy. The X chromosome carries a lot of the hereditary information. But here we can see that there are 3 chromosomes present at the 21 marker. So this is trisomy 21, which again is a very common form of Down syndrome. Now there are other kinds of Down syndrome, but trisomy 21 is one of the more common ones. There's an extra chromosome here when there shouldn't be, when there should only be 2. Now sometimes we can see changes in the X or the Y chromosome resulting in too many X's or too few of them. Sometimes it doesn't cause any issues, and sometimes it causes major issues. So Turner syndrome is an XO, which means that the female only has one X chromosome. It doesn't have the 2 X's like it's supposed to. So usually they're short with a broad chest and widely spaced nipples, can be of normal intelligence, and function with hormone therapy. This is non-disjunction or oogenesis or spermatogenesis, meaning that there is only one X chromosome present and that other X chromosome is missing. Now sometimes we can get extra. Here's Klinefelter syndrome. In this case we've got an extra chromosome. So it could be an extra X, it could be this Y that's extra. But usually because the Y is present, this is going to be a male since Y signals the production of testosterone. They typically have an underdeveloped testes and prostate because even though they have a Y chromosome present, it's often hidden by the extreme female hormones. Some breast overdevelopment, again because of those extra female hormones that are present. Long arms and legs and long hands. They typically have near normal intelligence unless we have way too many X's. And if that happens, then the intelligence can get a little messed up. And no matter how many X chromosomes are present, the presence of a Y chromosome renders the individual male. So that Y chromosome is what determines if an individual is male or female. there's something called the SRY gene. The SRY gene is the gene that is on the Y chromosome that basically determines if it is male or not. And sometimes the SRY gene gets deleted and results in an XY female. So even though the Y is present, it's not showing, this individual is not showing any male characteristics because the gene that's on the Y chromosome is not triggering male representation within the body. This usually is a lack of testes determining factors which play a role in the male genital development, and the presence of the SRY gene determines maleness. The SRY gene actually takes up the majority of the Y chromosome. So even if a Y chromosome is present, if we don't have this particular gene, then the individual is not going to show those male traits. Some abnormal sex chromosome numbers. So again, usually the sex chromosome is determined at number 23. In this case, the X is missing, so this would be X0, which would be Turner syndrome. Here is Klinefelter syndrome, which has 2 X chromosomes plus a Y, so it's got that extra chromosome there that is still giving this organ or this individual male appearances, but with some extra female hormones as well. Now, there are very many different kinds of mutations that can take place. We can have deletions, duplications, and translocations. A deletion is when something is taken away completely. One or both ends of a chromosome broke off, something is gone. 2 simultaneous breaks leads to a loss of internal segments, and so we can lose information with a deletion. We can also have a duplication where too much information gets repeated, and that can cause issues. And then a translocation is basically taking a chunk and swishing it, so almost like we saw with crossing over, but it's not with the homologous chromosome. So it's a chromosome that is not similar to it. So this is messing up the genetic code, whereas with homologous chromosomes, it didn't really matter because the chromosomes were similar anyways. But if we're doing this with a non-homologous chromosome, that's going to mess up the genetic information as well. An inversion, it occurs as a result of 2 breaks, and it gets inverted and basically flip-flopped as well. The internal segment is reversed and kind of goes backwards instead of going the right direction. And genes occur in reverse order in the inverted segment. So it's the same one, but instead of reading from this direction to this direction, it reads from this direction to this direction instead. And this is important because when a DNA is trying to make a protein, it's reading the protein incorrectly then. So we can have some syndromes that are a result of deletions and translocations. Williams syndrome is the loss of the N chromosome 7, which contains the gene for elastin. Children have turned up noses, wide mouths, small chins, and large ears, and this is part of what gets deleted in this process. With translocation syndrome, myeloid leukemia, a blood cancer caused by translocation between the chromosomes 22 and 9. Alagille syndrome is a translocation that happens as well. So here we can see how these work. A deletion is this one is taken out. A duplication is we have DE and it duplicated to DE. An inversion, it was going A, B, C, D, and now we go A, B, D, C. It's switched and went the wrong way. And translocation is, again, just cutting a chunk off, almost like crossing over, but in this case with non-homologous chromosomes. Here are just some cases of instances that can happen. In this case, with a deletion, you can see how the changes in the individuals, they have the wider mouth and a distinctly different shaped head than an individual who could have the one without the deletion. Leukemia is a process where there are extra white blood cells and not enough white blood cells. All right, well, that's all the information for this chapter, and I'll see you guys for the next one. As always, if you have questions, don't hesitate to ask. Bye, guys.